cell membranes are one of the most prominent structures that we can ever find in both cells and cell organelles. They play an important role, which primarily is to regulate or rather separate the internal environment and the external environment. It's either of the whole cell or the internal environment of the cell organelle. Now, the membranes that we're talking about, we have that one big cell membrane that is found particularly in both plant and animal cells. We also have internal structures which possess cell membranes. Those structures are called membrane-bound organelles. And membrane-bound organelles, they have a cell membrane which performs a function that is similarly to that of the main cell membrane. With me here is just the general structure of that cell membrane. And those membrane-bound organelles include the endoplasmic reticulum, the Golgi apparatus, the mitochondria, the chloroplast, the nucleus, and some others are even double membrane, just like the mitochondria. We also have the nucleus. It's even double membrane bound. However, we want to start by looking at the structure of the cell membrane in general. It's actually applicable to all these structures. And after that, we look at the functions in detail. But generally, like I said, the function of the cell membrane primarily is to separate the internal and external environments. Also, the other bigger part that the cell membrane is playing is that it regulates substances that are moving inward and outward of the cell or the cell organelle. And that type of movement is controlled simply because the cell membrane is partially permeable. Partially permeable simply means there are some substances that are allowed to move into the cell and other substances that are not allowed to move. Okay, so look, if you look carefully at this diagram, there are parts that are supposed to be labeled. We're going to start by labeling all these parts, and then from there we actually go into detail in terms of the function. The most prominent thing generally in terms of structure is that the cell membrane is what is in a structure that is called the fluid mosaic model. Why is it called the fluid mosaic model? Simply because it has got the fluid part. The fluid part is regulated mainly by the phospholipids. These are the parts that are in red with black tails. They're called phospholipids. They are the ones that give the membrane its fluidity. However, the degree to which it is fluid is actually determined by the tails, which we'll talk further when it comes to the zoomed part there. And the mosaic idea of it is that there are haphazard arrangement of different structures that are others are on top, others are on bottom, and it's creating a haphazard arrangement. That means it's mosaic. It's alternatively called a scattered arrangement. Okay, so to begin with, let's talk about this red part here. This red part here is called the phospholipid. This is basically the phospholipid. But what part exactly is the phospholipid head? And then here is the phospholipid tail. We also have other prominent structures, the ones in blue. These are proteins. They are particularly divided into two. We have what we call integral proteins, and then there are what we call peripheral or sometimes extrinsic proteins. Integral proteins are proteins that span the whole along the whole membrane structure, and then extrinsic or rather peripheral proteins, they are found at the ends, either inside the cell environment or outside the cell environment. For example, this one is a peripheral protein. We also have this one, which is this one is a protein, but if you look at it carefully, it's a protein that is attached to another side chain. And that side chain, what we call it, this is a carbohydrate chain or a short carbohydrate chain that is attached to a protein. So as a result, this type of protein is known as glycoprotein. That side chain is a carbohydrate side chain. Carbohydrate side chain. We also talk of this here. Now, in this structure here, it's called a glycolipid. Why are we saying it's a glycolipid? The carbohydrate side chain is now attached to the lipid, to the phospholipid head. So as a result, we change the name to become glycolipid. And we also have a different way of classifying our proteins. Proteins can be classified based on functions. Most of the proteins that are found along the cell membrane, they are responsible for transport. These ones that have got a space here are known as channel proteins. This is the zoomed structure here, which will explain more in detail, but basically these are channel proteins. Again, we have proteins like this which have no channel but are also carrying part in terms of transport. We can also even see them from here. This is a carrier protein. Both carrier proteins and channel proteins are involved in transport. We explain more in detail how they're involved in transport. Again, we have these blue small embeddings that are found in between. These are called cholesterol. What cholesterol does, we are also going to mention its functions. Yeah, so from this diagram, I think you can see that we have managed to label all these parts based on the generalized structure. Another generalized fact about the structure of the phospholipid is that it is basically idealized to be ranging, when it comes to the size, it is 7 nanometers in diameter. And also you can see that it has got two layers, a layer of phospholipid that is facing the inward environment, whilst the other layer is facing the external environment. As a result, two of these layers result it to be called a phospholipid bilayer. And again, you should also ask a question to say, why is it some of these proteins, they are attached? What makes them to be attached to the cell, to the membrane? There should be forces acting. So there are two main forces that act so that the proteins remain attached in this structure. The first force that is embedded in this, it can be understood if I come here and further explain the forces that are found when it comes to the head and the tail. The phospholipid is divided into two: the hydrophilic head and the hydrophobic tail. These are the main structures. So this part here, I will say, is the hydrophilic head, and this one is the hydrophobic tail. These two are responsible for creating certain types of forces. The hydrophilic head forms ionic forces, or rather they are called water loving, which means they attract ionic molecules as well as water molecules. This also enables this protein. As you can see, there is a linkage here. There's a linkage here, which we call ionic interactions. These ionic interactions can be basically understood when you know how the structure of a protein looks like. It has got an ionic part, which enables charges to attract. According to the principle, like opposite charges attract, these form an ionic interaction. However, the hydrophobic tail is water repelling. Let me just label here and say water loving. We also have water repelling. Since the hydrophobic tail is water repelling, it simply means it is not in line, or rather it does not attract ionic molecules. As a result, it forms hydrophobic interaction. Now that we have looked at the general structure of the cell membrane, now we need to look at the function of each and every structure of these parts of the cell membrane. Remember, each structure of the cell membrane is just not there for display. It's performing a specific function that aids to the overall efficient function of the cell membrane. We'll start with the most important components, or rather the biggest function of the cell membrane, which we said it regulates the movement of substance into and out of the cell. It is partially permeable, meaning to say there are some substances that are not allowed. Which substances are allowed and which are not? So it allows substances based on two things. Number one, substances are allowed based on size. And then number two, they are allowed based on charge. When it comes to transport based on size, we have two further categories there. There are small molecules. We have small molecules and large. Small molecules, they can simply pass through the cell membrane because they are small enough. They cannot be hindered. They are very small. Small molecules like water molecules, they pass freely. So they pass through the cell membrane by what we call passive transport. Passive transport. And large molecules, they need bulk transport. They can't just pass, they need bulk transport. What is passive transport? Passive transport is just passing through the cell membrane like freelance, no disturbance, no stoppage. And of course, the large molecules are passing by bulk transport. Now, when it comes to transporting molecules based on charge, remember we have the hydrophilic head, which we said is water loving. So it attracts ions. But generally, the membrane, we have non-ionic molecules and ionic. Non-ionic molecules, they pass by passive transport. They pass by passive transport. But ionic substances, they can be attracted by the hydrophilic heads, but they are not then able to pass through the cell membrane because they are then repelled by the hydrophobic tails. So as a result, they need proteins. Specifically, we talked of two proteins. We said there is carrier proteins and there is channel proteins. Carrier proteins and channel proteins are responsible for transporting ionic substances. So ionic substances are undergoing a type of transport that is known as facilitated. Facilitated, it means it requires a medium. Okay, in this structure of the channel protein, which I've zoomed in here, ions that can pass through here, mainly we have sodium plus, Cl chloride ions, potassium ions, phosphate ions. All these channels, they pass through. What enables them to pass through is because these channel protons, they are water filled. They are filled with water such that they can easily be attracted to the channel as they pass through. They are water filled, and this is the channel that they use. Carrier proteins are transporting stuff like glucose. That's where we have the GLT, the GLUT carrier protein that is found on cell surface membranes. This is number one function, which is transport. Again, the second function of the cell membrane is we talk about, when it comes to the cell membrane, we talk about enzymes. Cell membranes have got functions whereby the proteins found on the cell surface membranes are acting as enzymes. We have two main processes that I will talk about. We have the process of what you call this, the process of respiration. The process of respiration is simply having enzymes that are embedded within the membrane, just like the ATPase enzyme. We also even have the complex 1, complex 2, all those enzymes. they facilitate a process that is called electron transport chain. Those are protein molecules that are embedded within the cell membrane. Even the GIT, we look at the GIT system, the small intestines, they have microvilli projections embedded with enzymes as well that are responsible for converting food into their smallest particles, like converting amylase enzyme, converting carbohydrates into smaller molecules. Now, this is the other function. The third function that we're going to look at, which is facilitated by cholesterol. Cholesterol is embedded in this. What it does is it maintains fluidity. It maintains fluidity here. Cholesterol maintains the fluidity. Maintains the fluidity. That means there are animals that are staying in very low temperature environments. Those animals are at risk of having an inefficient cell membrane. Why? Simply because the temperatures are very low. But those animals have got high levels of cholesterol in their cell membranes, which enables the fluidity of the cell membrane to be maintained. Again, the next function, which is called recognition. Cell membranes, if you see these structures like this carbohydrate side chain, which is said is the glycoprotein, they have got these carbohydrate side chain that enable recognition of the cell as part of the tissue or part of the body system, which we call antigens. So these ones are acting as antigens. Basically, what this does is that it enables such that when antibodies or white blood cells are passing through trying to recognize foreign cells, they will actually try and test the antigen to see if it's originally originating from the cell. Again, from the cell body, rather, the body system. Again, the other function, which is, what else do we have left here? Apera. No, apera katika zomira that. So this is hydrophobic interaction here. Hydrophobic interaction. And of course, what does it do? It then keeps the protein to be held tight to the cell membrane. Looking a little bit further more on the structure of the phospholipid, we said it has the hydrophobic head. The hydrophobic head is made up of a lipid, or rather, let's say a phospholipid. This is phospholipid, but specifically the phosphate group. The phosphate group. The tail then is made up of a lipid part, which is called the fatty acid. The fatty acid. Let me just take you a little bit back to organic chemistry. You know that fatty acids, or rather lipids, they are actually alkenes. They have two types. There are those that are saturated and those that are unsaturated. Unsaturated, they have got double bonds. Those double bonds, they are very, very important significance when it comes to the structure of the membrane. The more unsaturated the membrane is, the more it is more likely to be fluid. Therefore, it is always advisable that you eat unsaturated foods so that the membrane structures of your body are actually going to be more fluid-like. Basically, if we look at all this whole structure, these are the levels of the structure of the cell membrane. Now we need to look at the functions of the cell membrane. All right, guys, so we have managed to do the structure and function of the cell membrane. And I just want you to hit up the subscribe button, follow us on all our channels, as well as to see the other video, which is linked to this one, which is cell membrane transport systems.